Imagine if someone is teasing and embarrassing you, but instead of getting embarrassed, you think of a witty comeback and attack them instead. This, in the most basic sense, is how one of the most revolutionary scientific discoveries works. It's called CRISPR Cas9. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So how does CRISPR work? In order for CRISPR to work, bacteria must have survived an infection by a bacteriophage during the lytic cycle, so it contains viral DNA. This viral DNA will be cut into fragments. In the chromosomal bacterial DNA, there is a CRISPR locus which has CRISPR repeats. Between these repeats are the viral DNA fragments from each infection added at the 5' end. RNA polymerase transcribes this DNA into mRNA, which is cut into cRNA, including a CRISPR repeat and a guide RNA. Meanwhile, transactivating CRISPR RNA or tracer RNA is complementary to cRNA and binds with the Cas9 enzyme to form a Cas9 complex. Therefore, if bacteria is infected by the same virus again, then a section of the injected viral DNA will flip and attach to the guide RNA so Cas9 can make a double-stranded break. Many Cas9 complexes float around the cell, continuously cutting DNA and acting like an adaptive immune system.
So why is the CRISPR Cas9 system such a revolutionary discovery? Well, currently scientists have been able to manipulate the CRISPR Cas9 system by creating a specific guide RNA that targets a specific sequence in the human genome. And by doing so, they're able to make a double-stranded break in the DNA. And then they inject corrected sections of that DNA that through homologous recombination can create a fixed section that fixes the mutation. Now, in an article called CRISPR the Disruptor by Heidi Ledfor, scientists at MIT have already injected the CRISPR-Cas9 system into mice, and they've corrected a mutation associated with a human metabolic disease called tyrosinemia. It was the first use of CRISPR to fix a disease-causing mutation in an adult animal. The CRISPR-Cas9 system can be used for so much. We could use it to modify every cell in a zygote, potentially cure cancer in a localized area. Or we could use it to edit crops so that they could grow in areas like deserts. Also, we could use CRISPR-Cas9 to create a gene drive in animals like mosquitoes, which carry the gene for malaria and wipe it out. But of course, with all this potential comes some concerns in ethics. For example, what if we introduce the CRISPR-Cas9 system and it targets a gene that we didn't want it to? Goodbye, gene for blue eyes. Or what if we use the CRISPR-Cas9 system so much that it wipes out diversity, and the gene pool becomes so similar that the human population becomes too susceptible to new diseases? Overall, CRISPR-Cas9 is like a prescription medicine. It has one big potential to cure and change the world, but side effects may include. So I suggest that for now, we find some directions and learn a little bit more about how this miracle genetic medicine works before we put it in ourselves. Thanks for watching.